Hey everyone, Mr. Murphy here. I'm going to talk about Gregor Mendel and his contribution to genetics. So Mendel was a monk in the 1800s who lived in a monastery and basically was tending different plants. And he became curious about the way in which the pollen being exchanged between the plants would lead to a genetic mix of the two parent plants in the next generation. And looking at these different types of pea plants, he saw a whole number of ways in which there were variations between them. And he developed experiments that sort of broke down what was the likelihood of two plants of a particular trait having a next generation of plants that would either retain or not show those particular traits. It was pretty remarkable at the time because, again, this is like sort of the beginning of genetics. This is before anybody knew about DNA. Obviously, people had known for thousands of years that, kids tend to look like their parents, but there was no understanding of any sort of biological or chemical explanation for that phenomena. So here was this monk in the middle of the 1800s who really kicked off this whole thing, and that's why he's still known as the father of genetics and why Mendelian genetics is still an absolutely essential and important part of all biology classes. So looking at the pea plant, here's what made that kind of unique. The pea plant has a flower which is fairly enclosed, and there's different structures to a flower which enable the process of reproduction. So there's the stigma, which this is marked as sort of like the female component of it. This has a tube which leads to the ovum where the egg cell is found, and then there's the anther which produces the pollen. Now, it's kind of weird to think about this, but what pollen is basically a capsule which contains the plant sperm cell. Kind of gross when you think about, like, we breathe in pollen all the time, but obviously plants are very different than animals in terms of how this sexual reproduction occurs. But it's important to realize that it is sexual reproduction. It is the combination of two different plant organisms to produce a new generation that's unique genetically. So we have female and male components to this. Because of these structures and the accessibility of these structures in a pea plant, he was able to basically brush the male components, the anthers, which have the pollen, and deliver those to another flower, to the female component of that. And in that flower, he's able to remove those anthers, so that way you prevent what's called self-fertilization, which is a way in which a lot of plants actually can have pollen from that plant actually cause fertilization in that same plant, which gets a little bit strange in terms of the fact that that's still sexual reproduction, even though it only involves one individual. It does cause a new genetically unique organism, but it gets a little complicated in sort of the exact breakdown of how the chromosomes are unique and all of that. We don't need to get into that today, but it's kind of interesting stuff. So Mendel's doing this again in the 1800s and stumbles upon the fact that he begins to unlock sort of the way in which genes or our different physical traits seem to disappear sometimes, but then pop back up in a future generation. And obviously people had been aware for a long time that, kids tend to look like their parents. But what they weren't aware of was the way in which those traits are coming from genes which are always going to be there, but are not necessarily going to show up except in certain circumstances. And to help explain that, here's just the simplest form of his experiment, where we have a tall pea plant and a short pea plant. And what he did was he crossed these. So he took pollen from one, applied it to the other, and these were true tall plants. So ordinarily these tall plants would only produce tall plants, and these short plants would only produce short plants. Okay, but when you combine them, what he found was they produced all tall plants. Now, that seems like that's not a particularly interesting thing, but the really interesting part of this was when he then took those all tall plants and he allowed them to self-fertilize. So pollen and egg from the same plant. What he found was that suddenly some of the next generation produced short plants. So while it seemed like this generation here had lost, the shortness ability, when he allowed them to self-fertilize, it sort of came back. And in fact, it came back as roughly 1/4 of the total total number. And in fact, there's well, it keeps getting covered up. The exact numbers from Mendel's experiment are listed there: 787 tall, 277 short. So the guy's looking at over 1,000 pea plants, which is, again, pretty incredible. You got to put yourself in sort of the time frame of this. But what he found was that you get this 3-to-1, 3 tall for every 1 short coming out. And what this showed through our later understanding with Punnett squares and how we represent different genotypes and different alleles and different versions of these genes which are contained in these organisms, what it showed was that if we use these letters to represent the genotype, we saw that these tall plants had big T, which means big T codes for tallness and little t codes for shortness. So these true breeding tall plants would be big T, big T. Remember, they have 2 alleles because they got 1 allele from each of their parents. So the short plants then have 2 little t's, and the only way in which a short plant will appear to be short is if it has 2 of the little t's, because these are what we call recessive traits, which means that they don't show their expression unless you have 2 of them. Or another way of putting it is if you have at least 1 of the dominant allele, then you are going to show the dominant trait. And so when these are bred together, we can set up a Punnett square that shows how at the top here, the tall ones can either put forward a big T or they can put forward a big T. And the short ones can either put forward a little t or a little t. And then we just sort of put these next to each other and across the top, and when we go down and we go over and we see, okay, big T, little t, so that's one possibility of the children of this plant. And then big T, little t, that's another possibility. Big T, little t, that's a third possibility. Big T, little t, that's a fourth possibility. Not that unique in this case. All 4 out of 4 possibilities of how these things could combine lead to the same thing. Big T, little t, which we call heterozygous because it has 1 dominant and 1 recessive. So that's why we end up with all tall, because as it turns out, if you have even 1 dominant allele, you show that trait. You show the phenotype of tallness. Phenotype being like the physical manifestation of those genes, so physically what you look like. But then when we mix these with themselves, we end up changing our Punnett square, because now each of these plants can put forward either a big T or a little t. And so if we put one of those plants across the top and one of those plants on the side here, and then we think about what the different possibilities are, if the first plant gives a big T and the second plant gives a big T, then you end up with big T, which would be a tall plant. If the first plant gives a little t and the second plant gives a big T, then you get big T, little t. If you do the first plant big T, second plant little t, then you get big T, little t, or both could give little t's. And this Punnett square here shows us why we end up with 1 out of every 4 of these plants being short, because there is a 1/4 chance of the result of mixing these 2 plants together. It's a 1 in 4 chance that there'll be 2 little t's and therefore be short. One last thing just to address about this, between the big T and big T little t, you would have no way of knowing which one's which, because there's no like half degree of tallness in pea plants. There are some traits where having 1 dominant and 1 recessive can lead to something in the middle, or lead to sort of a coloration that might be halfway, but this is not that particular type. When it's 1 dominant and 1 recessive, you just show the dominant. And so in these plants, you would have no way of knowing which one is the big T and which one's the big T, little t, until you take them and you set them up with another plant and you run another round of reproduction, and then you see what results. And that's how you could sort of deduce the ability of these organisms to be carrying that recessive allele, because if you're heterozygous, then you're going to be tall, but you don't know whether you carry the gene for that recessive allele. So that's a little bit about Mendel's experiment and a little bit of an introduction to the Punnett squares, which we'll be doing next. Certainly if you have questions, let me know, and I hope that is helpful.